Hello, good morning everyone. Myself Pushpa. So till now we have discussed about the history of chromatography, definition of chromatography and various types of chromatographies, and already we have discussed about the column adsorption chromatography. So what are the different stages of column adsorption chromatography? What are the requirements we have seen already? First of all, column chromatography, it is divided into two types, that is column adsorption chromatography, column partition chromatography. In the previous class we have already gone through the column adsorption chromatography. Both column adsorption and column partition chromatographies are same. In both cases we will use column only as a chromatographic bed, but the stationary phase whatever we are using is different from column adsorption chromatography, whereas in column adsorption chromatography we will use solid stationary phase, whereas in column partition chromatography we will use the liquid stationary phase. That is the only main thing difference between the partition and adsorption. Because of that difference, the mechanism of separation is also varying. In column adsorption chromatography, the separation will takes place based on the adsorption. That means with how much affinity the sample is adsorbed on the stationary phase and it is eluted along with the mobile phase will be explained in adsorption chromatography, whereas how much affinity or with how much solubility the sample is solubilized in mobile phase and how much solubility is there for the sample with the stationary phase, based on that solubility the sample will be get partitioned into the mobile phase, or based on the solubility the sample may get partitioned into the stationary phase. This principle is happening in the partition chromatography. These are the two differences which are happening in the column adsorption and column partition chromatography. First of all, we will see what are the differences between column adsorption chromatography and column partition chromatography. Then we will go with the explanation regarding the column partition chromatography. Here see. So first of all, I already told you column adsorption chromatography and column partition chromatography both are column chromatographies only, but the main differences and similarities are, see, the main similarities are both are the chromatographic techniques only based on the separation only. Both are used for separation. Both are using column as a stationary phase or chromatographic bed. These are the similarities, and both are used for the separation of the compounds, and both can be used for separation of solid samples, liquid samples, gaseous samples. That means whatever the component which we are going to separate, either the component is solid or liquid or gas, column adsorption chromatography we can use, column partition chromatography we can use. These are the similarities. Coming to the differences, what are the differences between the column adsorption chromatography and column partition chromatography? Both are used for separation of components only, but the main principle is column partition chromatography is used or it is worked based on the principle of partition, whereas column adsorption chromatography is based on the principle adsorption, and whereas partition it is based on the principle partition. Partition means you already know what is meant by partition coefficient. Partition coefficient is nothing but, normally the general definition of partition coefficient is how much sample is partitioned in the lipid phase and how much sample it is partitioned in the water phase. Best example is when we are passing a sample, either it is a paracetamol drug or any other chemical compound, into oil and water phase. Oil and water both are immiscible. They will not be mixed with each other, miscible with each other. If whatever component which we are adding to that oil and water, if that component is having more solubility in the oil, then it will get solubilized in the oil and it will move on with the oil. If that sample is having more affinity with the water or solubility with the water, then it will solubilize more in the water and it will move on with the water. That means based on the solubility of the component which we are separating, maybe it will be partitioned into the oil or maybe it will be partitioned into the water. That is called as partition coefficient. That means partition coefficient is nothing but the partitioning of the sample either in the liquid phase or in the aqueous phase. is called as, based on its solubility, is called as partition coefficient. Same definition we have to apply here in the partition chromatography. Here, there, how the partitioning is happening is based on the partitioning of the sample in the oil phase and water phase. Here, the partitioning is happening based on the with how much solubility the sample is partitioned in the mobile phase, and with how much solubility the sample is partitioned in the stationary phase will leads to the separation. If the sample, for example, I am going to separate the paracetamol drug. That means in the paracetamol drug there will be paracetamol pure component, some other excipients like binding agents, lubricating agents, coloring agents, preservatives. So I want to separate all those excipients along with the pure paracetamol. In the drug, some components, maybe paracetamol will have more solubility in the stationary phase, and other excipients like lubricants or preservatives maybe have some solubility in the mobile phase. So whatever component which is present in that paracetamol, if it is having more solubility with the mobile phase, it will move on with the mobile phase. It will get partitioned into the mobile phase. If the component which is present in the paracetamol mixture is having more solubility with the stationary phase, then it will partition into the stationary phase. So based on the solubility of the component which is present in the paracetamol, the components will be partitioned into either stationary phase or into the mobile phase. So the main principle which is happening here is partitioning of the components of the mixture based on the solubility. That is happening in the partition chromatography. Whereas adsorption chromatography, already you know, based on the binding interaction or affinity, the sample will get absorbed on the stationary phase or on the mobile phase. We have already seen that in the previous lecture. That is the main difference between the adsorption chromatography and the partition chromatography. The other differences are here in partition chromatography, the stationary phase we are using is liquid, and also the stationary phase here we are using is the liquid, and the mobile phase is already liquid only. Whereas in adsorption chromatography, the stationary phase we are using is solid, and the mobile phase we are using is the liquid. And coming to the separation, I already told you the separation is based on the partition, and here the separation is based on the adsorption. These are the similarities and the differences between the column adsorption chromatography and column partition chromatography. Next, coming to the main principle which is happening in column partition chromatography is, I already told you, the main principle is partition, in which the partition is happening based on the solubility. See, in this picture we can see how the stationary phase, I already told you, in column chromatography, column partition chromatography, we will take the stationary phase in the liquid form only, and we will load the sample along with the mobile phase. The green color one is the sample along with the mobile phase, and this blue color one is the stationary phase. So whenever we have first of all we will take the stationary phase into the column, and then we will add the sample along with the mobile phase, and after that we have to left over this column for some time, and then there will be happening interactions based on the solubility of the components which are present in that sample. Maybe the components of that sample, if they are having more solubility with the stationary phase, it will get interact with the stationary phase. If it is having more solubility with the mobile phase, that is pink color one, it will get soluble with the mobile phase and move on with the mobile phase. So we have to left this system like this for some time for the better solubility and better interactions. After that there will be weak interactions and strong interactions. That means if the sample is having more interaction with the stationary phase, it will strongly interact with the stationary phase. If it is not having that much interactions, it will interact weakly with the stationary phase. Based on that interactions, they will elute in different timings. If it is, for example, if the component in the sample is having more interaction with the stationary phase, it will get solubilized more with the stationary phase, and it will take more time for coming out. But if the component which is present in the sample is having very less affinity with the stationary phase, that means it is not soluble with the stationary phase, so it will not spend more time with the stationary phase. That's why it will come, or it will elute fast, but the sample which is having strong interaction with the stationary phase, that is here you can see the brown color, it is having more affinity with the stationary phase or more solubility with the stationary phase, that's why it is having more interactions with the stationary phase, that's why it will take more time and finally it will elute very slowly. Same like adsorption only, partition also separations will take place, but here stationary phase and mobile phase are liquids, whereas in adsorption the stationary phase and mobile phase are solids and liquids. Okay, this is happening in the column partition chromatography, and the principle behind this is, I already told you, we have to take the immiscible stationary phases and mobile phases, both are liquids, but both should be immiscible, both should not get miscible. And the stationary phase whatever we are using is, especially you know already we have discussed in the column adsorption chromatography, frequently used stationary phases are silica gel, alumina, magnesia, all those things, but in this column partition chromatography we will use those silica only, but how can we call it as a liquid stationary phase? What will happen here is, we will take the solid silica gel only, but that solid silica gel we will cover with the liquid surface. That means in that silica gel we will mix the liquid solvent. You can see here in this procedure the immiscible solid surface, that is silica, is covered with the liquid surface which is acting as a stationary phase. That's why here we are calling the stationary phase as a liquid phase. That means it is not completely liquid. We will take the solid silica gel, but we will mix that solid silica gel with the liquid solvent or liquid mobile phase. That's why here the stationary phase, which one we are taking, act as a liquid stationary phase, and the components which we are going to separate we will get mixed with the mobile phase, and then we will pour it into the column which is already having the stationary phase like silica with liquid solvent. The separation is happening based on the partition. How the partition will happen? Partition will happen based on the solubility. So the formula is K is equal to Cs by Cm. K is nothing but the partition coefficient. How the partition will happen? Based on how much solubility happening. So Cs by Cm means how much concentration of the sample in the stationary phase, and how much concentration, Cm means how much concentration of the sample or the solute in the mobile phase. See, Cs by Cm means concentration of the sample or solute in the stationary phase and concentration of the sample or solute in the mobile phase. How much concentration is present in the stationary phase means, if the concentration of the sample in the stationary phase is more, means it indicates the sample or the solute is having more solubility in the stationary phase. If the concentration of the solute or the sample is more in the mobile phase, means obviously it was indicating, it was denoting that whatever the sample we are separating, it is having more solubility in the mobile phase, and that's why it got more partitioned into the mobile phase. Understood? The main principle is partition. It is depends upon the solubility. K is equal to Cs by Cm. And then next, coming to the types of column partition chromatographies. First of all, we have seen what are the similarities between column adsorption and column partition chromatography, and dissimilarities or differences between the column adsorption and partition chromatography, and then we have seen the principle behind the column partition chromatography. And coming to the next one, types of column partition chromatography. One is liquid-liquid partition chromatography and liquid-solid partition chromatography. In this type of partition chromatography, here it is this liquid-liquid partition chromatography mainly we can use for paper partition chromatography. Here the liquid-liquid partition chromatography means, in the name itself it was telling that we will use the stationary phase liquid and the mobile phase liquid. Mostly these are useful for the samples which are having different partition chromatographies. That means for example, in different samples there will be different solubilities. For example, we have taken one mixture which is containing 10 components. All the 10 components may not have same solubility in the stationary phase or mobile phase. Some components maybe will have more solubility in the stationary phase. Some components may have more solubility in the mobile phase. Based on their solubilities, the sample components will get partitioned into stationary phase or mobile phase. And the next type is gas liquid chromatography. In the name itself, we are denoting that. In the previous one, liquid liquid chromatography. In that, both stationary phase and mobile phases are liquids. Here, gas liquid chromatography. That means the mobile phase will be gas and the stationary phase will be liquid. Best example is GC. You know, gas chromatography. Obviously, gas chromatography means the mobile phase, whatever we are using, will be in the gas form. The mostly commonly used mobile phase gases are helium, hydrogen, neon. And the stationary phase is liquid itself. And one more thing we have to take care about this type of gas liquid chromatography is whatever sample we are going to separate. If it is in gas form, well and good, we can use this gas chromatography. But if the sample which one we are going to separate is in liquid form, then you have to convert the liquid sample into the gas form, and then we have to use this gas chromatography for separation of that sample. For example, if it is in solid form, if it is in liquid form, you have to convert into gas form. Same like that, if it is in solid form, first of all, we have to mix with some solvent, and then we have to convert into liquid form, and then finally, by heating, we have to convert into gas form, and then that gaseous sample we have to send into this gas chromatography for separation. These are the two types of partition chromatographies. One is liquid liquid partition chromatography, gas liquid partition chromatography. And coming to the next one, I didn't mention the procedure which is happening in column partition chromatography because we have done already with the column adsorption chromatography. The process, everything, stationary phase, mobile phase, preparation of the column, packing of the column, and elution or development and the detection, everything whatever is happened in the column adsorption chromatography, same will happen in the column partition chromatography also. And the factors affecting column efficiency or dimensions of the column: particle size of the adsorbent, nature of the solvent, temperature of the column, and pressure. Dimensions of the column, you already know, I already discussed in the column adsorption chromatography. Same like that, even in column partition chromatography also, 100:1. That means if the length of the column is 100 centimeters, then diameter of the column it should be 1 centimeter. Then it will be a better and efficient column for good separation. Okay, those length and diameter dimensions also very important for the good separation of the column chromatography. Whereas particle size of the adsorbent, if the particle size of the adsorbent is very high or large particles, means maybe the samples may got struck into that particles, or be in between the spaces between those large particles of the stationary phase, maybe the sample will get holded. So there should not be more particle size and there should not be very less particle size. Regular or uniformly placed particles of the stationary phase should be placed in the column, because if the particle size of the silica or stationary phase whatever we are using is uniform and uniform size and regular size with 6 to 20 micron sized, means there will be good separation of the column chromatography. And the nature of the solvent, whatever the solvent or mobile phase which one we are using, it should be inert. That means mobile phase should be inert. It should not react with the sample, or it should not react with the stationary phase, or it should not react with the column which one we are using. That means that much inert it should be, but it should have the property of solubilizing the sample. If it is not get solubilized, if the sample is not solubilized in the mobile phase, it can't go through the column or the adsorbent and there will be no better separation. It should act as a solubilizing agent, but it should not react with the sample or stationary phase or the column which we are using in this separation technique. And one more thing very important is the solvent or mobile phase whatever we are using, it should be very less viscous. If it is high viscous, there will be no good flow. If the mobile phase is not flowing goodly, or if it is not flowing thoroughly, there will be no proper elution, and it will not make the sample also to get eluted fastly. That's why the solvent or mobile phase whatever we are using, it should have very less viscous property. And the next one is temperature of the column. Mostly already you know. Coming to the solubility properties, the temperature will increase the solubility. For example, best thing is if you are mixing the sugar with the water by using the spoon, it will take more time whenever for example, if you are keeping it in on the stove and if you are heating and stirring means it will take less time, because of that heating, because of that increase in the temperature, it will obviously increase the reaction between the molecules of the sugar substance, and then with the water also there will be strong interactions, there will be strong solubility. That's why if you are increasing the temperature, there will be more solubility of the sample with the stationary phase and the mobile phase, and then because of that more solubility there will be more partitioning occurs and leads to better separation of the sample. Obviously even with the pressure also you have to increase the pressure. More pressure is there means there will be more separation, that means there will be more flow of the sample between the stationary phase and mobile phase. Because of that good flow, there will be more interactions happens for the sample with the stationary phase and mobile phase, there will be better separation. These are the factors which are affecting the column efficiency. If these factors are very good, there will be, that means if the factors are which are affecting the column efficiency are in a optimum level, in a good level, there will be better separation. And then coming to the next thing, applications like that of column adsorption chromatography, we can use column partition chromatography also for different applications. It will be used for separation of so many compounds, so many chemical substances, so many drug samples, and also so many phytochemicals also. Example, it will be used for the separation of glycosides, amino acids, lipids, proteins, alkaloids, and also it will be used for removal of so many impurities present in different drug samples, impurities present in different phytochemicals, etc. And also it is used for the separation of active constituents from the plant extracts, and also it is used for the estimation of drug. For example, in the paracetamol 500 mg drug sample, in that paracetamol tablet, you know already there will be paracetamol, but along with that it will have some excipients also. So how much amount of pure paracetamol is present in that drug along with excipients, if you want to determine, we can use this column partition chromatography. Best example is a determination of how much percentage weight by weight of strychnine present in the syrup of ferrous phosphate with quinine and strychnine. That means among this syrup which is containing strychnine, quinine and ferrous sulfate, pure amount of strychnine how much it is present, we can determine by using the column partition chromatography. And also this column partition chromatography already we have discussed in column adsorption chromatography also, that column adsorption chromatography and also this column partition chromatography we can use for the separation of enantiomers, diastereomers, those are stereochemical compounds, we can separate by using this column partition chromatography. And also this column partition chromatography is also used for the separation of tautomers and different types of racemic mixtures and different types of chiral compounds also. So these are the uses. And also it is also used in DNA and RNA sequencing. In the genetical applications also we can use this column partition chromatography, and also determine the pollutants present in food products and beverages. Best example is, for example, when you gone through noodles, we will use Maggi noodles and Yippee noodles. In that there will be some other components also which will be hazardous or harmful to our body. So what are those degraded or impurity compounds which are present in that food products, we can identify by using this column partition chromatography. And also we can use this column chromatography, column partition chromatography, to isolate polar compounds and non-polar compounds. That means polar compounds means which are having more polar nature, OH group containing compounds, and also to separate non-polar compounds also we can use this column partition chromatography. And also for the isolation of metabolites of different biological products. Metabolites means you already know, whenever one particular biological compound, best example is cholesterol, it will get metabolized into steroids, bile acids, other hormonal compounds, vitamin D, so many are there from metabolism of cholesterol. So what are those metabolite compounds from different biological compounds we can identify by using partition chromatography. Best example is, whenever we gone through the urine analysis, what are the different metabolite products we can see by using the column partition chromatography. And coming to the advantages, this is most commonly used separation technique for separation of different components, and here we can use very less sample quantity for separation, and also we can use different types of mobile phases. That means ethanol, methanol, polar solvents, non-polar solvents, so many wide variety of mobile phases we can use for this type of chromatography. And coming to this one, here in column partition chromatography there is preparative type also. You already know the difference between analytical and preparative. Analytical means the technique which we will use only for separation, whereas preparative means we will separate and also we can collect that separated compound. So that's why this column partition chromatography we can also use for preparative type. That means separated compound we can collect finally also. And also for analysis, automation is also possible and easy for this column partition chromatography. And coming to disadvantages, already you know it is a time consuming process. That means it will take more time for better separation. If the component or mixture is having so many samples or so many components, then it will take more time for separation. And also person is that means one person should be there while this process is happening. So those are the disadvantages. And one more main disadvantage is more amount of solvent will be consumed for the process, and automation makes the technique more complicated. We can do the automated, that means we can make this column partition chromatography which is manual, if we can make it as a automated, but when we are doing this as an automated process, it will be more complicated and also costly. These are the things which are happening in the column partition chromatography. Okay, thank you.